Welcome back. This is part 3. This is the last of the lectures of the cell cycle. We have introduced the basic idea. We've talked about interphase. Interphase is the biggest part of a cell's life, and also includes DNA replication, and it includes a little bit of getting ready, those centrosomes dividing and replicating. I want to point out here, because I think it's a good place to remind you, cell theory states that all life is made out of cells. Cells are the smallest unit of life, and cells always come from other cells. We talk about the cell cycle, or in prokaryotes, binary fission, because this is a basic concept of life. Cells make other cells. This is also all the way back to the origin of life. One of the patterns we see that suggests that all life came from the original cell. We think we're related because we do these processes, whether we do binary fission or mitosis, we do them with similar proteins. We do them in a similar pattern. That similarity suggests common ancestry. And then the differences suggest evolutionary pressure over time. Let's go. I always joke with this part that I could literally teach this in 30 seconds, but it wouldn't be very good. But it actually wouldn't be bad. It's a little cycle. I've narrowed it down to 4 steps. Be aware that as you go on and you take cell biology and you take your graduate courses in biology, there are more steps here. These are the 4 big concepts. I think one of my grad students told me one time they had 19 steps, 19 big stages in mitosis. When you draw this and you're going to take it on to med school and you're going to use your organizer for the future, leave space, because there's a lot more going on than what I'm telling you. I'm just giving you the general outline here. We're going to divide the mitotic phase, the M phase, but don't really call it that. That sounds weird. The mitotic phase into 4 different stages: prophase, metaphase, anaphase, and telophase. PMAT. At this point, I make everyone in lecture yell PMAT. And we PMAT, and you PMAT, and you PMAT. And it sounds like a swear word, but it's not. So if you can just get this down today, before this video is over, just PMAT. And PMAT, not tired and you're exhausted and you're stuck at home, PMAT. If you get that down, you got most of it, you've got the framework built in your head already. I tell my students every year this. And about a year or 2 ago, I was literally running down the street, and the mailman pulled up next to me, and he leaned out. You know how they drive on the wrong side of the car? In the U.S., they drive on the other side of the car. The mailman leaned out and he was like, PMAT, Dr. Beal, PMAT. So I must have had him at one point as a student. But he remembered, and you should remember too now. PMAT, and that's half the battle. Woo, that was too fast. I am going to give you some things that happen. I am really interested in the chromosomes and what they're doing. I'm less interested in the cell structures inside. I don't know if my head's over this. I'll move it to the side in the next video, or in the next slide. But I want it to keep track, even though it doesn't matter for this cell, I want to keep track of mom and dad's chromosomes. This is our spider mite chromosome. I've simplified it a bit here. It has a big one and a little one from mom, and a big one and a little one from dad. During S phase of interphase, these chromosomes replicate it. They are now stuck together at their centromere. This chromosome has 2 sisters. They are in the beginning of prophase. They start to condense. They wrap up tighter and tighter and tighter and tighter. I'm not going to do the whole thing because it'll be a 4-hour job. They wrap up tight, and they actually condense so much that they are visible sometimes with a light microscope. At this point, they're so wrapped up into nucleosomes and then ultimately into coils and then supercoils They're wrapped up so tight that the cell cannot use them right now to make proteins, but it can organize and move them around very easily. The nuclear envelope is still there at the beginning of prophase, but the centrosomes start extending out these tubes. They're called microtubules, tiny tubes. These microtubules form the mitotic spindle. The reason this was named spindle is, and I don't have these at home, I don't do this, but when people used to make yarn or thread, really, you'd get a sheep and you'd take the sheep and you'd shave it. You get all the wool, and then you card the wool. You get all the burrs out of it, and then you take it with what's called a spindle, and you twist it and twist it and twist it until it becomes thread. And the way that the spindle drops, it helps you twist it very tight, and it becomes a very strong thread. All that wool twisted together. When people first saw this process occurring, and you can see it with a light microscope, they thought that this centrosome looked like a woman using a spindle spinning threads. And so they called this the spindle fibers, because they look like threads are being thrown out from the centrosome. Now these threads need to get all the way to the kinetochore handles on the centromere, but there's a nuclear envelope in the way. So as prophase continues, the nuclear envelope starts to break apart, and the spindle fibers grow. They reach out towards the centromeres, and the kinetochore handles, those little protein wings that shoot off the centromere, shoot out from the centromere, and they reach for each other. I drew these little purple blobs are my kinetochore handles. Prophase ends when the spindle fibers reach out and attach to the kinetochore handles, and we start metaphase. By the end of prophase, go back one slide, all the organelles are gone. They're there. They've just been broken up and shuffled to the side. The nuclear envelope is gone. Everything's been pushed away. Everything's been packed into vacuoles and pushed out of the way, because the biggest thing that needs to happen now is we're going to spend a lot of time trying to organize our chromosomes. Metaphase starts when the spindle fibers connect to the kinetochore handles. Metaphase ends when the chromosomes are lined up in a straight line across the center of the cell. That we call that center line the metaphase plate. It's kind of like the equator of the cell. And like the equator, it's an imaginary line. It's a line we draw in between two poles. In this case, the poles are the centrosomes. The centrosomes have thrown out spindle fibers, migrated to opposite sides, and now the spindle fibers start to tug on the chromosomes until they line up in the center. This takes a long time. These can't see. Nobody can see here. So it takes a long time for these chromosomes to line up in the middle of the cell. As they do, once they line up on the metaphase plate, metaphase is over. So, bob back. Prophase is all of the getting ready. The organelles disappear. The chromosomes condense. The spindle fibers reach for the centromere. The kinetochore handles start to appear and start to reach for the spindle fibers. Metaphase starts when everybody comes into contact. Spindle fibers come into contact with kinetochores. Metaphase ends when everybody's lined up. Metaphase immediately becomes anaphase. And it's really this fast. If I go back to metaphase, anaphase. Anaphase is the shortest stage of mitosis. As those chromosomes line up on the center, as soon as they're on the metaphase plate, an enzyme is released that cleaves means cuts, that cleaves the cohesion proteins and results in disjunction. Disjunction means pulling them apart. Disjunction sounds like something is wrong. We want disjunction. We separate them, and the chromosomes start to move to opposite sides. Now, when you see this in real life, if you're watching this happen, it looks like almost like they rubber band apart. They don't. Those little dynein proteins that we talked about walk them away from each other. So I'm like here, and the chromosomes migrate to opposite sides. But there's so much APP involved. There's so much energy involved that it happens like whew, and now they're to the opposite sides. Now here's a question for you. We went from chromosomes with sisters, we lined them up on the metaphase plate, and now we separated them. Did our ploidy change? Because now look, I got 4 chromosomes. 4 chromosomes. Do I have new sets? No. Ploidy doesn't change in mitosis ever. We get around this, and anaphase is a little bit funky here. That's why we add more stages when you go on into other classes. But at this point, this is one set of information, 2 sets of information that's going to go on to a cell by itself. This is 2 sets of information that are going to go on to a cell by itself. And so we say the ploidy has not changed. We have just separated sister chromatids into chromosomes. Anaphase starts when the cohesion proteins are broken, and it ends when the chromosomes reach the opposite side of the cell, a matter of seconds in some cases. These are chromosomes. These are chromosomes. We now have 2 sets of chromosomes, 2 complete sets of chromosomes that are going to represent the genetic information of one cell and another cell, and they are identical to the cell we started out with. After anaphase is telophase, PMAT. Telophase or telophase is where we are going to kind of run prophase backwards. We need to put the cells back to the beginning. Remember, in every cycle we've done, we've had a part where we restore the beginning molecule, or we get back to the beginning of the cycle. Telophase is us getting back to the cycle, except now we have 2 identical daughter cells. Each cell has to go back to interphase G1, which means it has to put itself in working order. We're going to form 2 nuclear envelopes. They're going to be smaller because we're forming them from the remains of the parent cell. But we have 2 nuclear envelopes, 2 sets of organelles are going to form. For the mitochondria, some will go this way, some will go that way. And these 2 cells should be genetically identical. As telophase occurs, cytokinesis occurs at the same time. But cytokinesis, the pinching in the middle, is not part of mitosis. It's its own thing. Cytokinesis is about the cytoplasm dividing. The reason it's its own thing is that some cells, like your muscle cells or fungus cells, don't necessarily go through cytokinesis at the end of mitosis. They create multinucleate cells with many nuclei, but they're not separated by cell membranes. So cytokinesis, although common, is not required as part of this process. It's its own process on top. In animal cells, the cell pinches in on the side and pops apart. This is done with actin, the same type of proteins that help your muscle contract, and some of those microtubules cause it to pinch in and separate. And it does it in a way so that the cytoplasm doesn't ooze out. In a plant, plants can't do that because they have a cell wall. Fungus can't do it because they have a cell wall. In plants, as the cells go through telophase, and their new nuclear envelopes start to form around their chromosomes, they start to generate what's called a cell plate. This is a lot of sugar. And as this sugar is released in between, it builds the framework for a wall. The plant then builds the wall on top, and ultimately I end up with 2 tiny plant cells. And where do we go from there? It's a cycle. So we're going to go back to the beginning. Each cell, daughter cell, then goes back to G1. It grows. It lives its life. It makes proteins. And then someday, while it's grown up and living its own best cell life, it gets a signal from the body that says, hey, cell, are you ready to divide? And the cell says, I am. And the cell enters the S phase. And during S phase, it replicates its DNA. It checks its DNA. If all's good, it goes into G2. G2, the centrosomes replicate. And we go into mitosis. Prophase. Chromosomes condense. They become visible. They become wrapped. The nuclear envelope disappears. The centrosomes start to throw out the spindle fibers, and they move to opposite sides of the cell. And as the spindle fibers touch the centromeres of the chromosomes, metaphase starts. Metaphase starts on as we attach the spindle fibers to the centromeres. It ends when everybody lines up on the metaphase plate. Anaphase, cohesion proteins are broken. And now we start telophase. The nuclear envelope returns. The chromosomes decondense. They go back to their working form. Organelles come back. And cytokinesis occurs concurrently. And then we go back to the beginning again. This is a cycle. The cell cycle is surprisingly a cycle. I'm going to leave you off with one question to think about and compare. I think you are maybe watching this before exam 1. If not, this is good practice for the final and exam 1 too. Exam 1 also. I'm going to have you compare and contrast prokaryotes and eukaryotes. We talked about this when we compared and contrasted them a month ago. So here, which occurs in both binary fission, metaphase, or DNA replication? And I'll see you in the next lecture. We'll get comfortable with PMAT, because in meiosis, we're going to PMAT. And I'll see you soon. All right, bye-bye.